Greetings of the day. Today we will be seeing about the second week of development, which includes the implantation, a very important 6 marks question or in the short answer section. Second one, it is the formation of the bilaminar disc, and also the next content of today's video is the precaudal plate. So these are the three topics which we are going to discuss in the today's video. So the second, these all includes the second week of development. So first of all, let us discuss about the implantation in detail. So implantation basically it is divided as basically it is defined as the process in which the embryo gets implanted into the uterine endometrium. So before that one, last week in the last video we have seen about the formation of the blastocyst. So this blastocyst basically contains the inner cell mass, and then first of all it contains the inner cell mass. So this inner cell mass and then the outer cell mass, that is the another one. And we have already learned that the outer cell mass is giving rise to a structure called as the trophoblast, or the outer cell mass is also called as the trophoblast. So these are the basically two layers or the two parts of the blastocyst. And now the process of implantation is defined as the implant inward grow or the implantation of the embryo into the uterus. That is the end of the first week. Actually what happens is that that may end of the first week, that is on the seventh day. On the seventh day what happens is that the polar trophoblast or simply the trophoblast, its nature is a sticky in nature. So trophoblast is sticky in nature, and what happens is that it gets attached to the columnar epithelium. So it gets attached to the columnar epithelium of the uterus, and specifically saying, which is an important MCQ, that is the posterior uterine wall. So the implantation or the sticking of the trophoblast usually occurs on the posterior uterine wall, and then this endometrium of the posterior uterine wall is broken down by the proteolytic enzymes secreted by the polar trophoblast. Actually proteolytic enzymes, they are secreted by the trophoblast. So let us get chart sort of thing. Okay, so endometrium is broken down by the proteolytic enzyme secreted by the proteolytic enzyme secreted by the trophoblast. And then on the eighth day, the next day, what happens is that the trophoblast differentiates into the inner trophoblast. Let me write it. So it divides or it differentiates. So it's not division, it is differentiation. That is into the inner cytotrophoblast inner cytotrophoblast and outer syncytiotrophoblast outer syncytiotrophoblast outer syncytiotrophoblast. So syncytiotrophoblast, the word meaning is that there will be no cell membranes, and instead it looks like a bunch of nuclei, whereas cytotrophoblast has got a well defined cell membrane. And now what is happening is that on the next day, that is on the ninth and the 10th day, on ninth and the 10th days, on the ninth and the 10th days, the procedure will be happening is that the syncytiotrophoblast will be undergoing the mitosis very fast, and in this process its cells lose their adjacent cell membranes. So that is called as the multinucleated protoplasmic mass, called as the syncytiotrophoblast. And what is happening is that this syncytiotrophoblast further it secretes more enzymes so that the more erosion of the uterine endometrium takes place. And what is happening is that now it comes and now the endometrium is in the secretory phase. Now actually during this all process, the point to be understood is that the uterine endometrium is in the secretory phase. Secretory phase. So in the secretory phase, what happens is that there occur some changes which are all concluding, which are called as the decidual reaction, which is another important question and also a concept to be understood. So decidual reaction. So let us understand in clearly. So to sum up before going further, let us keep in the simpler words. That is the first of all, the on the seventh day, the polar trophoblast of the blastocyst being sticky in nature gets attached, and the erosion starts place due to the action of the trophoblast. And now the next day, the trophoblast is differentiating into inner cytotrophoblast and outer syncytiotrophoblast. And on the ninth and tenth day, further it undergoes rapid mitosis to grow, and finally it forms the multinucleated protoplasmic mass which is termed as the syncytiotrophoblast. And as it is further secreting the proteolytic enzymes, what happens is that there occurs a concept and a phenomena called as the, which is termed as the decidual reaction, which is called as the decidual reaction. So an important concept and also a question which is very important. Now what are the characteristics of this decidual reaction is that first of all the intercellular substance increases and its vascularity also increases subsequently. And now the stromal cells have got a separate name. Stromal cells, they have got a separate name. Why? Because due to the enlargement and becoming filled with the glycogen and also lipid, now they are termed as the decidual cells. Now they are termed as the decidual cells. And now after this one, the endometrial glands they become elongated, and after that one, endometrial arteries also gets elongated and become more tortuous. Now importantly, the lacunar spaces, now importantly, the lacunar spaces starts appearing in the trophoblast. Lacunar spaces appear appear in the trophoblast, and they are called as the, or they are called, they form the large lacunar spaces. And now on the 11th and 12th day, on the 11th and 12th days, what is happening is that the lacunar spaces enlarges, the lacunar spaces enlarge and further, and most importantly enlarge and communicate with each other, enlarge and communicate with each other. And now now the spaces are there, and in between the spaces the cords of syncytiotrophoblasts are called as the trabeculae. And now what happens is that the trophoblast further forms the finger like projections into the columns, and they are called as the villi. So the there is a formation of the new structure called as the villi. So what is this villi? Is that villi are defined as the trophoblast forming finger-like projections into the columns. So now let me explain with the diagram. So this is the picture of the trophoblast, and instead actually these are the this is the trophoblast which are formed by the flattened cells, and here we are seeing the inner cell mass, we are seeing the inner cell mass, and they are almost nearing the endometrial cells, and as we have seen, this is lined by the columnar epithelium. These are lined by the columnar epithelium. So with the nuclei, with the nuclei, and then below that one, that is the, this is all the endometrium, and here you can see the arteries. So this is termed as the endometrium, uterine endometrium, and that is the trophoblast. And here this is the blastocyst cavity, also called as the blastocele. So this is the blastocyst cavity, and then this is the polar trophoblast. This is the polar trophoblast. This is a polar trophoblast. And what is happening is that in the further step, the same diagram. So but what is happening is that, let me differentiate. The trophoblasts are lined by the flattened cells. The trophoblasts are lined by the flattened cells. So these are the flattened cells, and then the inner cell mass as usually. So now what is happening is that the erosion of the uterine endometrium has occurred, and it is going further deep inside. So this is the columnar epithelium, and here the same. Okay, so this is the erosion, and here what is happening is that this outer layer is becoming rapidly dividing and forming a multinucleated mass, which is called as the syncytio trophoblast, which is called as the syncytio trophoblast. And in the further stage, this grows, and then so this is the, once again the same diagram here, the flattened cell mass, and here that is the inner cell mass, and here the outer structure, it is forming into the numerous lacunar spaces has formed, and the villi are formed. Okay, so these are called as the secondary villi. These are called as the secondary villi. Here what is happening is slowly the villi formation starts, and in the further process what happens is that they grow in the size, and they are called as the secondary villi. These secondary villi are going to form an important structural and functional unit of the placenta. Okay, so now here this is how the implantation occurs, the steps of the implantation occurs. So now let us see the same in a flowchart mode. So first of all, the hatching of the blastocyst takes place, or the formation, the hatching of the blastocyst, and now its attachment on the seventh day. Its attachment happens on the seventh day. The timelines are very important to be noted and to be remembered. And then erosion of, and the next level is that, next phase is that erosion of the uterine endometrium. Erosion of the uterine endometrium. And then what happens is that the next level is that embryo is completely embedded. Embryo is completely embedded. Is completely embedded, and that is on the 9 to 10 days. And after this one, next one, the defect in the uterine endometrium is epithelium, uterine endometrium, it is closed. That is on the 11th to 12th day. Fibrin clot happens and it closes the defect caused due to the erosion. and after that one, and on the completely 13th to 14th day, 13th to 14th day, what happens is that the epithelial lining is completely restored, that is simple columnar epithelial restoration, we can term it as a epithelial restoration takes place on the 13th to 14th day. So this is the simpler and easier way to understand the flow, to understand the different steps in the formation of the implantation. So after the implantation, the next step will be the formation of the bilaminar disc, which is the second part of this video, and next important step in the further development of the embryo. So that is the formation of the bilaminar disc. So the formation of the bilaminar disc, actually now to recollect once again, we have to understand that blastocyst is the spherical structure. Blastocyst is the spherical structure containing the outer cell mass, that is the trophoblast, which is flattened, and inner cell mass, that one, and that is called as the embryoblast, and there is a cavity, that is the inner blastocyst cavity. So now what happens is that on the day 8, what happens is that the cells of the embryoblast on the day 8th, on 8th day, what happens is that the cell mass differentiates into the two layers. So that is the embryoblast differentiates into the two germ layers. So the names have to be understood here: hypoblast, and then that is the hypoblast, and then the epiblast. Hypo means below and epi means above. So hypoblast and then the epiblast. So the hypoblast, that is the, so hypoblast, that is a layer of the small cuboidal cells. Layer of the small cuboidal cells. So the epithelial lining is to be importantly remembered. So that is the small cuboidal cells, usually asked in the MCQs. Hypoblast is lined by the cuboidal cells, and now it has got a separate name, that is called as the primitive endoderm. It is called as the primitive endoderm. And then after this one, next one, that is the epiblast, the second layer, that is the epiblast, and it is lined by the columnar cells. So epiblast, that is lined by the columnar cells, and then that is towards the embryonic pole, and now we can confidently say that is the embryo is the disc shaped structure. Embryo has become disc shaped structure, and what happens is the disc shaped structure, and it is made up of two germ layers, that is the epiblast and hypoblast, and now that is why we are calling it as the bilaminar germ disc. In the further development, what happens is that there will be the formation of the amniotic cavity. So now we will understand the formation of the amniotic cavity in the form of a diagram. So formation of the, so the same, the flattened cells lining, that is the trophoblast. Yes, this is the trophoblast with the nucleus. Yes, this is the nucleus. And now what happens is that there will be the inner cell mass at this end, and there is the flattened cells, that is the, sorry, cuboidal cells. So now what is happening is that there is a differentiation here. Low cuboidal cells, as we have seen in the previous one, low cuboidal cells. Now here, that is the above this one, that is the inner cell mass. That is the inner cell mass. And now inner cell mass. So let me draw with a different color. Yes, this is all, that is the inner cell mass, and here that is labeled as the trophoblast as usually. So the outer layer, that is the trophoblast. In the further step, what happens is that the inner cell mass, as we have seen in the previous slide, that is the ectoderm is changing into the ectoderm is changing into the columnar cells. So the same once again, the flattened cells, trophoblast. Yes, and now this is the low cuboidal differentiation. And now here what is happening is that the ectoderm is seen as the cells in the columnar cells. So here they forms into the columnar cell. And now what happens is that small amount of the fluid starts above to the cranial to the ectoderm, and that is called as the amniogenic layer, and that roof is lined by the amniogenic layer cells, and which further fills with a cavity, that is called as the amniotic cavity. So in the further development, what is happening is that, so once again, a flattened cells layer, okay, and then here there is the differentiation. So that differentiation, low cuboidal, okay, low cuboidal, and then that is the low columnar. Okay, so and then here what is happening, the roof is lined by the flattened amniogenic layer. Okay, so let me draw once again here. On this end is a bigger picture. Okay, and now here the cuboid, the low flattened cells, low cuboidal, and then that is the ectoderm, columnar cells, and now here the flattened cells, that is the amniogenic layer. So nuclei. So now let me label it so as to further reduce the confusion. So this is the amniotic cavity. This is the amniotic cavity. So that this is the same labeling, that is the trophoblast, and here this is the ectoderm. The upper one is the ectoderm, and the lower one, that is the endoderm, which that is why we are naming it as the bilaminar germ disc. And here this layer, that is the amnio amniogenic layer, amniogenic layer. Oh, so that is the layer of the amniogenic cells. So ectoderm is the second germ layer and endoderm is the first one. And now what happens is that deep this cavity is there, this is also will be lining by flattened cells like this. Okay, so now what happens is that a new cavity called as the primary yolk sac is formed. This is the primary yolk sac. This is the primary yolk sac which got formed, and after this one there is a separate name for this membrane lining the primary yolk sac. So name has to be remembered, that is the Husser's membrane. That is the Husser's membrane. So the Husser's membrane layer is the one which is lining the primary yolk sac. Okay, so now what happens is that the cells of the trophoblast, it further gives rise to a mass of the cells. What happens is that primarily separates the amniotic cavity and the primary yolk cell. So that is called as the extra embryonic mesoderm. So there is the extra embryonic mesoderm, and its classification in the next we will see in the by further understanding how the extra embryonic mesoderm is formed. So now what happens is that the second layer which is formed here. So now here in the second layer, that the further development, what happens is that we will be seeing the formation of the extra embryonic mesoderm. So now the first one we have drawn already, that is the trophoblast. That is the trophoblast, and deep to that one we will be seeing the embryo here. So here that is the bilaminar disc. Above that is the ectoderm, that is the columnar, and then here that is the endoderm. So here that is the ectoderm, and here that is the endoderm. And now here we will name this as the amniotic cavity. This is the amniotic cavity, and the below one, that is the below one, that is the primary yolk sac. Below one, that is the primary yolk sac. So now after this one, the Husser's membrane. So here this is the Husser's membrane. This is the Husser's membrane. And after this one, what happens is that this layer here, this is the extra embryonic mesoderm. That is the extra embryonic mesoderm. So this extra embryonic mesoderm, further in the further development, what happens? So before that one, let us try to understand why it is called as the extra embryonic mesoderm. So because this is lining outside the embryonic disc and does not form any tissues, and moreover what happens is that the cells of the trophoblast is giving rise, and that is the extra embryonic mesoderm, and this extra embryonic mesoderm doesn't give rise to any tissues of the embryo, and as it is lining outside, so that is why it is called as the extra. Now in the further stage, what happens is that numerous small cavities appears in the extra embryonic mesoderm, and what happens is that so all these cavities they coalesce or fuse to form a cavity called as the structure called as the extra embryonic coelom, also called as extra embryonic cavity, or most importantly chorionic cavity. And now due to this development, the primary yolk sac will be smaller, and now it is called as the secondary yolk sac. Okay, so now along with that, what is happening is that the extra embryonic mesoderm is also having two classifications, or you can say the extra embryonic mesoderm is having two parts. So now let us try to understand with the diagram. So this is the trophoblast as usually, and now what is happening is that so this is the extra embryonic coelom, and now deep to that one lies the bilaminar disc or the embryo, the division here. So above that is the ectoderm, and then below that is the endoderm. So here, so with the nucleus. So, and now here, now due to which there is an area where the coelom will not be extended, and that area is called as the connecting stalk. And here the same labeling here, that is the amniotic cavity. So here that is the amniotic cavity, and now due to which it became the smaller, and now we are calling it as the secondary yolk sac. The one of the two important changes happening in the further development. So the first one is the cavities occurring, and then due to which they are forming a structure called as the extra embryonic coelom. So this is the extra embryonic coelom, and that extra embryonic coelom is happening in the extra embryonic mesoderm, and due to which there occurred two different layers of the mesoderm. The one which is between the part lining the trophoblast is called as the somatopleuric. So the part lining the trophoblast. So now this is the trophoblast. This is the trophoblast, and the part lining it is called as the somatopleuric layer of the extra embryonic mesoderm, or also called as the parietal layer. So let me write it. That is the somatopleuric or parietal. So the parietal, this is the one which is lining the trophoblast. And now the another one here, this region that is the lining the yolk sac, that is called as the splanchnopluric layer. Splanchnopluric layer or visceral layer. Splanchnopluric layer or visceral layer. So the one which is lining the trophoblast is called as the parietal or the somatopleuric, and the other one you lining the yolk sac is called as the splanchnopluric or the visceral layer of the extra embryonic mesoderm. So now in the further step, let us try to understand that the somatopleuric layer or the parietal layer of the extra embryonic mesoderm is giving rise to a structure or the layer called as the chorion, and the splanchnopluric layer it is splanchnopluric layer of the extra embryonic mesoderm is giving rise to a layer called as the amnion. So the splanchnopluric it is giving rise to the amnion, and the somato that is giving rise to the chorion. And now in the further what is happening is that the chorionic cavity, in the chorion once again the cavity occurs, and that is called as the gestational sac or chorionic cavity, or also called as the extra embryonic coelom. So after this one there will be the formation of the precaudal plate. Let us try to understand that with the diagram. So next one, that is the chorionic. First of all, the first two steps are remaining. So that is the formation of the chorionic cavity. That is the chorionic cavity, and it is a large fluid filled cavity. So chorionic cavity, it is a large fluid filled cavity. Large fluid filled cavity surrounding the embryo, and it is the space which completely surrounds the primary yolk sac. So bilaminar germ disc and also the amniotic sac. Now what is happening is that the embryo, which is the bilaminar germ disc along with the amniotic sac and yolk sac, now it is suspended in the large chorionic cavity by the connecting stalk. So now let us try to understand by drawing the diagram. So here this is the outer layer completely, and in which there is the gestational sac. This is the gestational sac, and here now we will be seeing the two layered membrane. So that is that this is the amniotic sac, and here that is the secondary yolk sac, or simply the yolk sac. And now let us label it. So this is the gestational sac. This is the gestational sac or chorionic cavity. Gestational sac or chorionic cavity or extra embryonic coelom. And now here we will see this is the cytotrophoblast, and here this is the connecting stalk. This is the connecting stalk, and then so this is the embryo with the two layers. This is the embryo with the two layers, that is the amniotic and the yolk sac. And now in the next point we will try to understand the formation of the precaudal plate, which is an important question. So the formation of the precaudal plate. So in the precaudal plate, what is happening is that, so first of all, the bilaminar disc is becoming at one end a rounded area, so that becomes thicker, and that is called as the precaudal plate. So what is happening at the micro level or at the cellular level is that, so first let us try to understand the two layers. So first of all, that is the ectoderm. So after this one, below that is the endoderm. So that is the nucleus. Okay. So here that is the this is the embryonic disc as seen in the section, in the sectional view, in the sectional view. So here this is the ectoderm, this is the ectoderm, and here this is the endoderm. Yeah, this is the endoderm. Now in the at one end, or you can say there will be thickening forming, that is called as the precaudal plate. So that will define the cranial and the caudal end of the embryo. So in the rounded structure, so here this is the sectional view, here this is the rounded structure. So what happens is that here there will be the formation of the precaudal plate. So this is the precaudal plate. So due to which what we are calling is that the precaudal plates area where that is formed, that is called as the cranial end of the embryo, and the opposite will be the caudal end of the embryo, or the caudal or the tail end of the embryo. Now what is happening is that, so at the cellular level, now here we have seen, here it is ectoderm, that is the columnar, and endoderm, that is the cuboidal. Now what is happening is that the at one area, the, so this is the ectoderm as usually. Now the endoderm at one side, it will becoming the columnar, and that is the formation, that is the area where we will be seeing the formation of the precaudal plate, that is called as the formation of the precaudal plate. So now I will label this as the precaudal plate. This is how the precaudal plate is formed. So that is the area. Now it determines the central axis, as I have said, that is the cranial end and the caudal end. So this is how the precaudal plate is formed. So just to sum up actually, so first of all hypoblast and epiblast is forming, and then amniotic cavity and primary yolk sac is forming, and after that one extra embryonic mesoderm, and in them the cavities appear and they coalesce to form the coelom, and then formation of the connecting stalk, and formation of the amnion and the chorion, and lastly formation of the precaudal plate. Okay, so now here actually to end on an interesting note, let me write. So here actually in this video we have learned about the key developmental events of the second week of the embryonic development, and here all are forming, that is also called as the week of twos. Why? Because almost all the structures which are forming, they are double. So first of all the embryoblast is dividing into two layers. Embryoblast dividing into two layers, that is the epiblast and the hypoblast. Epiblast and then the hypoblast, and after this one formation of the two cavities. So cavities are the amniotic cavity and then the yolk sac cavity. Amniotic cavity and the yolk sac cavity, and extra embryonic mesoderm. Extra embryonic mesoderm, that is also dividing into two layers. Somatopleuric, somatopleuric, and then splanchnopluric. Somatopleuric and splanchnopluric. And then once again the trophoblast. Trophoblast is also dividing into two layers. So that is the cytotrophoblast, and next one, that is the syncytiotrophoblast. And then chorioamniotic membrane. So amnion and chorion. And villi are also primary villi and secondary villi. So if you observe the events which are happening, they are all like the twos, event of the twos, or we can call, that's why it is as the week of the twos. So with this I will end the video. Thank you very much.